Hello students, this is Vivid Botany. We are discussing now electron transport chain that takes place as part of light reaction. We know that photosynthesis takes place in two phases, one is light reaction, another is dark reaction. So here this question is extracted from light reaction of the photosynthesis. Light reaction takes place in thylakoid membranes and dark reaction takes place in stroma. So stroma is colorless homogeneous matrix of the chloroplast, whereas grana, thylakoid membranes arranged like stack of piles. So here I drew the diagram. These are called as nothing but the thylakoid membranes or the grana present in chloroplast. So what is the light reaction? How does it take place? That is the part of the question I would like to explain you here. You see, this thylakoid membranes are the main site where the light reaction takes place. So in this diagram, the so-called we have taken here the TS of thylakoid membrane. In this thylakoid membrane, the inner halo space is called as lumen, and outside thylakoid membrane that is called as stroma. Now, in the membrane we have here total 1, 2, 3 complexes. The first one is PS2, that is photosystem 2. This photosystem 2 will get excited. Actually, photosystem 2 contains chlorophyll A molecule that the reaction center gets excited when it receives 680 nanometers of red light. Here you see 680 nanometers of red light. What happens when it get excited? Usually when it get excited, it undergoes photoelectric effect. It loses an electron. So this electron will be carried by different electron vehicles, during which protons are pumped into the lumen of the thylakoid membrane. So this is what exactly the light reaction that takes place in plants. Another is PS1, photosystem 1. Photosystem 1 is having chlorophyll A molecule as the reaction center, where it gets excited at 700 nanometers of red light. So photosystem 1, photosystem 2, both are the reaction centers, but the excitation levels are different. PS1 at 700 nanometers, PS2 at 680 nanometers of red light. So in between the PS2 and PS1 we have here cytochrome B6F. It acts as a electron vehicle in between these two complexes. Another is PQ, that is plastoquinone. Plastoquinone is amphoteric ion carrier. Amphoteric means it carries not only the electron but also the proton. Proton is cation, electron is anion. Both are being carried by the plastoquinone. Another is here ATP synthase. This ATP synthase has got here at CF0, that is tail, and CF1, that is head. So the tail is towards the lumen, head is projected towards the stroma. And this is PC. PC stands for plastocyanin. This is plastocyanin, and it is copper containing mobile electron carrier, and it is present in between cytochrome B6 and PS1. Now let us see exactly how does it take place. See, first PS2 gets excited by beaming enough sunlight, that is 680 nanometers of red light. So when PS2 is beamed 680 nanometers of red light, it gets excited, ejects an electron. After losing the electron, P680 will become P680 plus. So remember, P680 plus is strong oxidizing agent. In biology, this is strong oxidizing agent. Strong oxidizing agent means it can get electron from other molecules without any difficulty. So this P680, after losing electron, P680 plus will get the electron from water molecule. Right. So water molecule is splitted here in the presence of sunlight by chlorophyll molecule is called as photolysis. This is called photolysis. It is discovered by Robert Hill, hence we also call it as Hill reaction. So after breakage of water molecule, the things that it liberates here, half O2, electron as well as the proton. So these electrons, one electron is accepted by PS2 and P6CIT plus will convert into just P6CIT or just PS2. Now the oxygen molecule, that is byproduct of photolysis, is released into atmosphere and enhances the oxygen percentage in the atmosphere. So this is called as photolysis, and photolysis of water is catalyzed by a multi-protein complex called as water splitting complex or oxygen evolving complex. So this water splitting complex contains 4 manganese. I wrote it here, you can see 4 manganese, calcium, chlorine, oxygen and tyrosine. What is tyrosine? It's an essential amino acid that we will see in another chapter called molecular base of inheritance. So calcium, chlorine, oxygen, tyrosine. These are the components of oxygen evolving complex or water splitting complex, which catalyzes breakage of water molecule and giving the electron to P6CIT plus to convert into P6CIT. Now, the electron that is lost from photosystem 2 is accepted by PQ. PQ is amphoteric ion carrier. It carries electron as well as proton. So while it is taking electron from PS2, it will take one proton from stroma. So it takes one proton from stroma, one electron from P6CIT, P6CIT or PS2 and converts to PQH2. It converts into PQH2, that is fully reduced plastoquinone. This PQH2 will donate, it will donate the electron to cytochrome B6F. It donates electron to cytochrome B6F and proton to the lumen. Do remember, plastoquinone will give the electron to cytochrome B6F and proton to lumen, right? And cytochrome B6F will give electron to PC. PC stands for plastocyanin, that is copper containing mobile electron carrier, and PC will give electron to PS1. But PS1 does not accept electron from PC unless it loses an electron. Why should it lose electron? Because when PS1 gets exposed to 700 nanometers of red light, then it loses an electron and electron deficiency is pulled up by PC, that is plastocyanin, right? And from this PS1, electron is taken to NADP plus, and NADP plus will take one proton from the stroma, one electron from PS1, ultimately gets reduced to form NADPH plus H plus. So this reaction, formation of NADPH, takes place in the catalyzed by an enzyme called FNR. FNR stands for FD, NADP oxidoreductase. FD, NADP oxidoreductase. So remember the passage of electron first from PS2, then to PQ, then to cytochrome B6F, then to PC, then to PS1 and at last to NADP plus. So ultimately, who is the electron acceptor here and who is the electron donor? I am asking a question here. Who is ultimate electron acceptor? I write here, what is ultimate electron acceptor and donor? That is the question here. So think about this. I will be giving you answer at the end of the video. Now look here, as PQ is pumping electrons continuously to cytochrome B6, PQ also pumps one proton for each electron into the lumen. For every one electron it carries, it pumps one proton into the lumen. So inside the lumen, the proton concentration increases. As proton concentration increases, the pH will be very low. Whereas outside the lumen, that is in stroma, there proton concentration decreases and pH will be more. Here I wrote here. In stroma, proton concentration is low and pH is more, right? Then protons will try to move from stroma into the lumen, but the membrane becomes here impermeable. The thylakoid membrane does not allow the translocation of protons from lumen to stroma, that is from high to low. Then how do protons will get into the stroma? That is the question here. Now, further process here, we find that is CF0, CF1 that I am showing here. CF1 is head, CF0 is tail. Together it is called as ATP synthase. So, this ATP synthase contains tail piece and head piece. Tail piece is CF0, head piece is CF1. So, this ATP synthase will allow the protons from lumen to stroma. So, while protons are passing through the ATP synthase, the head piece revolves with great speed and adds inorganic phosphate to ADP to form the ATP, that is adenosine triphosphate. Right. So, this formation of ATP is called as chemiosmosis. It is discovered by Peter Michel. Do remember, it is discovered by Peter Michel, for which he has been given the Nobel Prize also. Right. So, generally speaking, it is also called as non-cyclic photophosphorylation. Why should we call it as photophosphorylation? Because phosphorylation means addition of inorganic phosphate. Photophosphorylation means the addition of inorganic phosphate is taking place driven by sunlight. So we call it as non-cyclic. Why should we call it as non-cyclic? Because you know that electrons are electrons are being lost from PS2. Again, do the electrons are reaching again PS2? No. They are moving to ultimately NADP plus. So it is a not cyclic, but it is non-cyclic photophosphorylation. Right. Now, let us see the question here. In light reaction, plastoquinone, that is PQ, facilitates the transfer of electrons from, this is PS2 to cytochrome B6F complex. The first option is very much right. So, this is PS2 and this is cytochrome B6F. In between, what is present? PQ is present. Plastoquinone is present here. Right. So, let us see here what questions they have posed here. What is the ultimate electron acceptor? It is NADP plus. It is ultimate electron acceptor. Electron donor, this is water molecule. So, water molecule is pumping the electrons from the so-called thylakoid membrane to where ultimately to NADP. And NADP is getting reduced to form NADPH plus H plus. So, this is the non-cyclic electron transport or non-cyclic photophosphorylation. Hope you have understood it very clearly, I think. And you have any doubt, please drop a line in the comment box. Ask me, right? I am ready to explain you because the time is very less that I am shortening it. I am describing it very briefly. You want in detail discussion, we can do that. And one more thing is that my channel Vivid Botany, do subscribe my channel Vivid Botany. We will be discussing the majority of the questions, especially tough questions from the NEET 2020, 2021, 2019 also if time permits. Okay, so thank you very much and don't forget to subscribe channel and don't forget to write your opinion in the comment box. Thank you.